I'm on a journey to get better, and I want to do it with you. And I'm not just focusing on physical health, I'm focusing on everything: emotional wellness, spirituality, finances, relationships, and so much more. Every week, it will be my personal goal to bring us, the world's leading healers, experts, and game changers, to share groundbreaking secrets and tips to getting better in all areas of life. Getting better isn't easy, but it's a whole lot easier when we can do it together. Welcome to Better Together with me, Maria Menounos. Welcome back, everybody, to Better Together. Happy Monday. Hope you're having a great day wherever you are in this beautiful US of A.
Or if you're not in the US of A.
I was about to say, you didn't let me take my breath.
That was
I have some breathing
breath.
issues, Stephen. So if I'm clearing my throat, everyone, it's because I'm on like 18 new inhalers.
That make you forget about everywhere outside of the USA.
Yeah. So then I was going to say, wherever you are in the beautiful US of A or the rest of the beautiful globe, maybe if you're on Mars,
Yeah, what about the ISS, like the International Space Station?
perhaps you're on the moon. You might be on a base in Afghanistan. I mean, wherever you are.
That'd be awesome. I hope we get there.
You know I've done that, right?
Yes, I do know. Didn't you do that more than once?
No, I only got to do it once. It was quite costly because I had to buy kidnapping insurance. I lost my week of wages. It ended up costing more than a normal person's salary for a year for me to go for 8 days to Afghanistan.
Did
do
the
good
government
work.
pay for it?
No, I paid for it.
Oh,
Yeah.
I thought you went out there for the military.
It was part of a USO tour. It wasn't USO, it was another organization. But I couldn't take vacation. I had to lose my salary for that week. And then I had to buy kidnapping insurance and some other crazy insurance. And yeah, it was funny, but that was my contribution.
Can we do a live show from Afghanistan? Let's do it.
Oh my God, it was the greatest trip ever.
We got to get Uncle Sam to front the bill, but
Oh my god.
No. Yeah, it was really amazing. I got to drive the first MRAP in Afghanistan. Steven somehow, of course, knows what that is,
Of course.
right? Oh, his
I'm
eyes
pulling
lit
it up
up.
for you guys.
Okay, MRAP is like this big monster,
Whoa.
what would you call that?
It's like an all-terrain vehicle, military style, but it's got all sorts of deflection stuff on the bottom of it.
Yeah, the point is
Like a
you can
Humvee.
You
It's
can
supposed
drive
to
over MRVs and you can drive over roadside bombs. And
That's
things like
the point.
That.
Yeah, it protects you from roadside bombs. And so they let me drive it. And here I am in the middle of the desert driving like a psycho, and people are puking in the back because I'm like, taking bumps. And yeah, it was really fun.
Didn't you always want to? It was you who had this deep desire to be a race car driver, right? Wasn't it? Or Kevin always jokes about that.
He always calls me Maria Andretti. But I've always thought I was Mario Andretti. I used to drive my Camaro around Boston like a crazy person. I love driving, except in traffic.
Oh yeah.
But yeah, Afghanistan was an incredible trip. So if you happen to be in Afghanistan, hello.
A lot of people give those areas of the world, like Northern Africa and the Middle East, a bad rap. But I tell you, I went to Egypt when I was 18. And that's North Africa, kind of on the verge. And it was amazing. Driving up to the Red Sea, driving on the Nile. They're agricultural places. They have fresh vegetables, fresh fruit, fresh food. It's so awesome.
Yeah.
So if you can find a time that is safe, I really recommend the trip.
I'd love to go to Egypt.
Me too.
But I have to say, I am scared. I'm scared to go over those areas.
It's different as a guy. Honestly, it's very different as a guy.
Yeah. It's definitely my friends were like, let's go to Israel. I want to go to Israel, especially for Orthodox Easter and to see the light and the whole thing. But I still am scared.
Yeah, it's like the motherland for us.
Yeah.
I know. I have family in Egypt and in areas of Lebanon, and we just don't go. We just can't. It's just not worth it. The people who are in Cairo, they're safe. It's just, you just never know what's going to happen. You can't plan for it. So we just...
Yeah.
You got to be smart anywhere you go, though, honestly. You could go
Mexico.
Yeah, you could go to Mexico, or you
Not
could
allowed
go
to go there.
You could go to some places in California And
Wait.
like if you drive to the wrong place, what's up?
Hold on, Steph just said something. You're not allowed to go?
Not allowed.
Mom and Dad won't let you go.
Won't.
No way.
It's like the one thing.
Wait, does
Really?
that, you can't drive into it or you can't fly into it or anything?
Anything.
Oh.
Can't go to any area in Mexico. A lot of it has to do with my dad being a judge. They really suggest judges don't go.
No way.
Yeah.
Why?
Oh, because of the cartel.
Well, especially San Diego. That's like
Oh, because
half
that's
our-
his district.
Yeah, that's his.
Wait, so he's putting away these bad guys, and they're gonna want to get him.
No, they're going to want to get Steph and hold ransom.
Damn, Steph, that's scary.
Yeah, so I've never been, and it's one of
Wait,
those.
you've never been to Mexico because your dad's
Yes.
a judge in San Diego? Can we put a costume on you or something?
I've never been to Mexico either.
Guys, you have not lived unless you've had a margarita and chips and salsa
Tacos.
in Mexico.
Is that our AfterBuzz retreat?
I
Except
will-
we leave Steph behind?
Yeah. No, we're gonna smuggle
I'll
her
be
in.
at the border in Chula Vista.
We're gonna put her in a nun uniform.
There we go.
Sister Stephanie.
Steph Sabbath, a new episode of Better
It's
Together.
Stephanie, not Sabra, Steph
Stefan.
Sabropoulos.
Stefan Brabopoulos.
No more glasses, Steph. You're going to wear sunglasses.
Yeah.
Wow. See, who would have ever thought of that?
I know.
That's crazy.
And it's like the cheapest, most
Easiest.
Yeah, easiest tropical adventure. I guess not tropical technically,
Yeah.
but it's such a like paradise section. Everyone raves about it, but yeah, I can't go.
Wait, so when you had your like high school, what's that trip? Spring break.
Didn't go.
Where did they go?
Though everyone went to Cabo. In college, everyone went to Cabo. Never allowed to go. Yeah.
Wow. See, I was never allowed to go because my parents were too strict. But
That
Yeah,
sounds
and
like
it's
the
we
plot
had to
for
work.
the plot for Taken 4 is like Steph Sabra's not allowed to go to Mexico, but all her friends are going, so she sneaks over there.
My dad
and
is
gets taken.
obsessed. My dad knows all of Liam Neeson's lines.
wait, I had never seen Taken, and we were home in Connecticut last year. And Kevin went and we bought all this ice cream and all the fixings at this little like store down the street that has like all the old school stuff because it's like, vintage in a sense. And we got into the screening room in Connecticut and we watched Taken. And instead of laying back, I was like up like this the whole
such a good movie.
time.
So good.
And I was like, oh my God, how did I miss this? This is unbelievable.
I have a very particular set of skills.
Yes.
That's what
Skills
my
that
dad
make
says.
living a nightmare for men like you.
Yes. Oh my God. I want to be him so bad. And by the way, I think I am. I am the female Liam Neeson. Not that I would be
Let's
able to
make
like
a movie.
I'm not going to be able to do like all the
I
like
think you're the female
physical.
John Wick, though, because if somebody killed your dog, you'd kill everybody. You're John Wick.
I don't know
Wait.
if I would kill everyone. I think I actually I would hunt him down. Like here's the thing. Kevin knows this about me. I can find anything. You drop a needle in a haystack, I will find it. So when someone loses like their earring or their engagement ring, I'm like, I got you. And I have the most insane stories of people stealing stuff and I found them. And I mean, in a group of thousands of people on New Year's Eve, like I will find it or I will find you. Make no bones about it. Have no fear. Maria's here.
All right, Steph. We have to edit
Okay.
Liam Neeson next to her now.
All right. Yeah.
Well,
we're making this happen.
But what do you
Trust me.
do now that you find it? Like, I'd love the tip on this because Kevin
Oh yeah, I don't want to
got
tell
his
you my story.
Kevin
It's
got
a
his
little
phone
aggressive.
stolen last year.
Kevin got what?
Kevin got his phone stolen last year.
Oh yeah,
And
I went.
he
I
had
hunted.
Find My iPhone. Shows up to the front door. We don't have it.
Wait, you're missing a huge element of that story,
I'm waiting
Steven.
for you to tell me.
My ass hunted that person down. I went. Kevin sat like a coward in his car across the street. I went into this dilapidated neighborhood, terrified with signs that like say, keep out, we'll kill you kind of shit. Okay. It was like scary ass. I'm like filming. I go, here, film me going in. This might be my death scene. I might be murdered. And I went up and this woman came out and she was creepy as shit. And I was like, hey, so, and you have to be really like sugary sweet. I'm like, my husband lost his phone. Someone who lives on this property, I think might have accidentally grabbed it, thought it was theirs, blah, blah, blah. No. Rrr. And I'm like, no. But see, like, here's this Find My iPhone app, and this shows you where your phone is. And it's right over there in that little house back there. It looks like your guest house. And she's like, I don't know who lives there. And I'm like, but like you live here and it's all the same. And
The
And so
audacity.
finally, I got her to let me in, and I knocked on the door. And I'm like deep far out. And I'm sitting there, I'm like, Kevin better not be on his phone. He better be watching me as I'm going through this. So I went back, and this like guy was like, I don't have anything. And I'm like, maybe you took it by accident. Like, and I was trying to like appeal to like his sense of karma. And no. So I got out before anything bad happened.
But it was there.
It was 100% there.
Whoa.
There was just nothing I could do. Like, they were not copping to it. So what was I going to do? Kick him in the face, run in and try to search this scary ass hoarder's heaven? No. Not worth it.
I
Like,
did
certain
not
things aren't
know
worth
that.
it. Like, certain things aren't worth it. You
100%.
try.
100%.
It was a phone, though. It was a phone.
Certain things are not worth it. If it was my phone, maybe. But I promise you, Steph, if you were taken, I would find you. Promise. Like, you see how dedicated I am to my mom's health.
Yes.
And how
That's so
focused
true.
and detailed I am. You can't, like, I have, by the way, like, definitely in my past lives been an investigator, been CIA, FBI, soldier. There's all this stuff for sure in my past. And I did a past life regression, a mini one with Brian Weiss when I did my interview with him 2 years ago. And I remember seeing soldier boots. And I was like, damn, I knew I was connected to soldiers for a reason. So
I feel like
yeah.
you need to do some sort of role like this.
Yeah, it would be pretty cool.
You need to catch Jack the Ripper.
Wait, there was this company that reached out to me to do a show. They were going to do this like crazy ass show that was so dangerous, like super dangerous, where it was like, I have to go hunt people like this down. And I was like, wow, I get a lot of offers. This is actually really exciting. And I
Real
was like,
life?
yes.
Oh, no.
Real life investigative,
No,
like crazy.
I meant fictional, Maria.
Like, you'd have to wear a bulletproof vest.
Yes,
to-
like I'm not kidding. It was so intense. I'll find the email and I'll share it with you guys after. But and maybe for our Patreon members. But anyway, it was so intense. And I looked at Kevin. I go, Kevin, this is so me. I would kill at this. This would be so exciting. But I'm 41 and I don't know if I want to die like this anymore. Like, when I was 21, maybe 31, I was cool with dying like this. Like, I was like, whatever, it's all about the adventure. Now I'm like, I don't know. Don't know if I want to do that.
I'm glad that you're questioning this, Maria.
Yeah. Well, the project finally, it didn't seem like it's going forward. I don't think they were going to be able to get insurance.
I think there's massive liability things from what you're saying.
You know what? Naked and Afraid is pretty scary and has a lot of liabilities too. I don't know how they get past that. But
I would kill to film a movie with you, Maria, that is like Taken, but the Naked Gun airplane version of Taken, Leslie Nielsen's Taken.
I don't know that one.
You've never seen Airplane?
No,
You've never seen The Naked Gun?
never.
Same.
What, Maria? You have a door to be opened for your life.
Really?
Yeah, you are going to.
You think I'll really like it?
I think you will love these movies.
Really?
Oh my God, Leslie Nielsen is one of the best comedic actors of our time.
Okay.
Yeah, he
Never
passed
seen
away
anything.
5 years ago, but no, he's phenomenal.
Okay. So the comedy version.
A comedy version of Taken, like where you got to stay silent, you're sneaking up on the bad guy, you look to your left, and there's two dogs humping each other, and then the person gets alerted because you just hear that. You hear, "Ah!"
I love it.
Like that's the movie we
man.
got to film.
I'm telling you guys.
Oh, yeah, he looks familiar.
I would find you.
So we have about 10 minutes to get some takeaways in this intro.
You know what, though? Sometimes these things have to just be fun. You just got to go with it.
100%.
And, it is what it is.
I do love, because we talked about it in the last Patreon episode, but I'd love for our fans to know the story behind your laugh.
Mm-hmm.
That was amazing.
You're going to have to join Patreon.
Maria told us how she got her laugh.
Mm-hmm.
It was a big deal.
I'm surprised you never had heard that before.
Never.
But I will say, actually, first of all, I didn't get to our quote of the day. So I'm going to get to our quote of the day. When we love, we always strive to become better than we are. When we strive to become better than we are, everything around us becomes better too. And that is by Paulo Coelho, The Alchemist.
Yes.
I love that. And we are here with you every Monday, but we are also doing multiple new episodes for you guys on the Patreon.
Every Monday, every Tuesday, every Friday, you can get better together. Yeah, I almost messed it up.
Yeah.
So let's say we're talking to Dr. Jewett today.
Mm-hmm.
Well, guess what? tomorrow, you can get the after show where we go in depth on what we learned. Let's say you listen to the after show on Tuesday and you're like, man, it's getting towards the end of the week. I need something to listen to really boost me up. Like, I don't know. I need Maria's laugh. I need to know why she laughs that way. I'm going to tune into the Patreon episode because we release it every Friday.
Yeah, and we've been getting such great reviews on the Patreon episodes, which is so, so exciting. So thank you guys for your comments on all of the shows, because it drives us, it motivates us, it inspires us to keep on going. And the whole point of the Patreon is we really want to build out this community, and that's kind of how we can have more direct connect with you guys. And also, I mean,
you get the shows ad-free on Patreon too. So every Monday we publish it there too. You can subscribe to an RSS feed, just like your podcasting app. It makes it super easy.
And our whole goal is to make this completely ad-free. So let's get all of you guys joining the Patreon community. And once we are, in a bigger place with it, we'll be able to add more exciting stuff to it. So, and you can communicate with our guests. There's a lot of really cool stuff that is available through the Patreon. And so we want you to become a member and
give us feedback on the tiers. Because if there's things that you're like, oh, that's not worth it, or something like that, let us know. We want to hear you guys. We're new to this too. We're trying to get better together.
And if you help us, well, we will be better together.
So true.
So today we have an amazing guest on the show. I am so excited. Steph is like totally geeking out.
Oh my gosh, yeah. Who knew?
And by the way, Steven is going to geek out because she's
scientific.
Her name is Dr. Anahid Jewett. She is a woman on a mission to find a cure for cancer. For the past 26 years, she's dedicated her life to figuring out why cancer stem cells are able to survive in patients. And if you are like me and you hear stem cells and you think good, no, these are not the good ones. These are the bad ones. And so she really believes that the key is to focus on natural killer cells, and she has devoted her career to this, and she's created a really cool therapy that is having a lot of success, especially with pancreatic cancer. And she also created the first humanized mouse model with an intact human immune system that she can test these things on. So we're not just testing rats and hoping when it goes to clinical trials with humans that it will be similar in terms of the results. She actually can get real results, like on these humanized mice, which is so mind-blowing to me. And she's kind of like, she's such a gangster badass, but so quiet. Like, she's the featured speaker at all of these big research cancer events with, like, I won't name them, but like, the men that have like huge publicists and just figure out a way to just own it in a way and just be celebrated. She's so quiet. She doesn't even care about the celebration. Her full mission is to figure this out and to save people. She doesn't care if she makes money. She doesn't care if she's celebrated. I've never met someone who is so truly genuine about what she's doing. And so she inspires me, and she has been really helpful to me. We met her through Tracy Ryan, who was a part of Ricky Lake's documentary, Weed the People. And Tracy...
And we had her back in January, late January.
So her daughter has brain cancer as well, and Dr. Jewett, and they are working together on a few different projects, and they're doing a trial at UCLA. And so we were able to bring my mom in to get tested. So it was in March of this, was it March this year?
I believe it was.
I think it was, yeah. March of this year, we tested my mom's natural killer cells. And at that time, she was doing well, and we had, we were doing great. And she said, this is why your mom's doing really well. She has an abundance of natural killer cells, and the killing ability is high. So I was like, great, this is amazing information. Well, when my mom's tumor was resurfacing in mid-September, I brought her back for more testing. And it's great because we had a baseline from before. And so I'm actually going to find out today where my mom's natural killer cells are at, because we have a sizable tumor that we're dealing with now. the good news that I can share, which I haven't been able to tell you guys because by the time my day was over yesterday, I was not well and couldn't keep my eyeballs open. And so the good news is we have managed somehow to shrink her tumor a smidge. So the rate it was going at and growing at, I think the doctors were prepared for a massive disaster yesterday when they looked at her MRI. And I think they were pleasantly surprised. I mean, I know they were, to see that it actually looks smaller. And certain areas that we thought were other tumors are going away. So whatever we're doing is working. Because
that's amazing.
It should be like 4 times the size.
With the rate it grew at, because I remember one day it wasn't there, and then all of a sudden it was massive.
And then it doubled in 2 weeks. And so the fact that a month later it hasn't doubled and doubled, it's only actually gotten better, we were able to breathe a huge sigh of relief. The bad news is we are dealing with a massive tumor.
A brain tumor.
That, is dangerous, and we can't debulk it with surgery, and we aren't going to do radiation. And so we're having to make decisions today on what we're going to do with her treatments to continue on this process. So,
but how crazy that today we have Dr. Jewett here.
Yeah. So if you listen to the Patreon episode last week, I think we talked about the church that we went to.
Yes, we did.
And the healing experience we had there. And I firmly believe that is why this turned around the way it did. I believe that in the spiritual realm, that's what has taken over. And I think that in the physical realm, all of the other things that we've done with Mexico and with Cedars has been super amazing. So we'll find out from Dr. Jewett today about my mom. But my biggest thing is I want to teach you guys about natural killer cells. I want to teach you about her research. And I want to teach you about the success that she's seeing with this therapy so that you are educated. And, in China, this is like an everyday thing. People go in and they get natural killer cell infusions like we go to cheap massage parlors out here. So I think it's really important for us to be educated on this, and it's going to be the future. So without further ado, here is Dr. Anahid Jewett. So Dr. Jewett, thank you so much for joining us. Before we start, I neglected to mention that you are the professor and director of Tumor Immunology Laboratory, and you're also a member of the UCLA Tumor Immunology subgroup, so that people actually know where you are officially at.
Sure.
And that's where I met you, was at UCLA. So thank you so much. I would love to start with natural killer cells, because that's what we're here to talk about today.
Yes.
You've dedicated your life to studying natural killer cells and creating a natural killer cell therapy combined with your probiotic.
Yes.
Tell us what natural killer cells are and why they're our friends.
Okay. I'm not sure if you want me to talk about my background, because that actually will give you more insight as to why I started, working with natural killer cells. So I've been in the field for greater than 30 years. And my line of work, of course, is tumor immunology. So we study how immune system deals with cancer and how we can improve the function of different kinds of immune cells. So when I started my career, I knew that I wanted to work with this very specific cell called natural killer cells. I didn't understand at that point. I didn't know. Of course, we knew what these cells were and what they were doing at that point, but the knowledge base was not that developed at that point. So I had, a lot of opportunities to actually start learning more about the cells. As I, developed my projects in natural killer cells, I found out that actually these cells are incredibly important because, first of all, what they are doing, they're targeting the seeds of the cancer. No cell, even T cells or B cells or any other types of cells that we have, would be able to target these cells. And NKs are the only one that they do it. And that actually opened up the field significantly, because now we know that NKs work with T cells in a different way. They are not backup for the T cells. They're actually T cells may be backup for the NK cells.
What are the T cells doing?
Okay, so well, T cells, there's huge amount of work that has been done in the field on T cells, because first of all, it's very easy to get T cells. If we look at the peripheral blood and we look at lymphocytes, which are the cells that contain NK cells and T cells and B cells, we'll see that 70% of our lymphocytes are T cells. So there's huge numbers that one can work with, and so it's easy to work with, and more people went to that field because of the ease of use.
But do T cells combat cancer?
Yes, definitely they do.
So what's the difference between the NK cells and the T cells?
Okay, so I'm trying to come up with something very simple.
Yes.
Because
please,
because I actually teach this in a whole course. So basically, T cells can recognize tumors in a very specific way. NK cells do not recognize tumors in a specific way. They recognize cancer stem cells. But T cells can look at a tumor cell and say, well, this is a malignant cell, this is a transformed cell, so I have to get rid of it. So in a sense, they actually target differentiated cells, because differentiated cells have something on the surface called MHCs, or major histocompatibility complexes. And peptides, which are small proteins, basically, parts of the protein, gets expressed in the surface of these MHCs, and then it activates our T cells. And the T cell can say, okay, well, this is not self, and this is self. This is changed self, so we need to get rid of it. So in a sense, T cells will kill the differentiated cells, whereas NK cells kill the undifferentiated cells. And undifferentiated cells are a lot more aggressive than differentiated cells, because differentiated cells actually don't grow as fast, and chemo can kill the differentiated cells.
So it's like the Marines and the Army. They're all working together to kill the enemy.
That's right.
Okay.
That's right.
But the
That's a
NK
great
cells,
analogy.
but the NK cells are like the Navy SEALs, actually, because they're the ones going in for the real bad guys.
That's right.
Okay.
That's right.
I like that. So
Right.
you decided you were going to go into this field that no one was going into it because it was a little more difficult. It was uncharted waters.
Yeah, well, there were other people besides me, but they were looking at NK cells in a different way. I was more interested in the tumor aspect of it, whereas, lots of other people are studying what the biology of NK cells are. They're not so much interested in how NKs interact with tumor cells. So when I started working in tumor immunology, there were a handful of people that were studying cancer. But now we have significant numbers of people that are coming into the field, just because primarily based on a lot of the work that we have established in the field. Because, I remember when I started working in the area that I'm working, there were maybe one or two people, basically, trying to understand the role of NK cells, because the majority were with the T cells, and they thought T cells are the most important cells to eliminate cancer. Now we're realizing actually NKs, and we shouldn't discard the fact that T cells are very important too, because the combination of these two cells that keeps us healthy, basically, not only with cancer, but also during infections, during trauma, a variety of different things. So in terms of the, why I became interested, because, I remember maybe I was 4 or 5 years old, that my mom had this significant fear about this very mysterious disease that I couldn't understand. And at that time, I didn't understand what cancer was, right? I mean, you're a small child, you're just going based on the fact that someone is instilling that fear in you, and you start thinking, okay, well, what is this? I want to try to figure out what this is. And so since then, actually, that interest has been I guess, sown in me in order to be able to, to come up with ideas so I can help my mom. And interestingly, my mom actually was hypochondriac at that point because her mom had passed away from throat cancer. She was worried that she is going to pass away from the same kind of cancer. And so, I was, 13 or 14, I decided this is where I'm going to go. At first, I thought I'm going to be an oncologist because I want to treat. But then soon after I realized, really, oncologists are not going to be able to do much because the field is not grown. It hasn't been developed. We don't have enough treatments in the field. So that was when I started thinking, okay, well, the research is going to be the way that I will, go. But interestingly, one event from, your childhood can determine the path of your life. And for me, I think that's what started, but I think, God put that mission in me. And,
For sure.
And now I feel that this is something that I have to do. I'm here in this world to do this.
So where are you at with natural killer cells?
So, I published about 150 publications in the field.
That's it? You've only published 150 articles in this field? You are such an underachiever, Dr. Jewett.
I believe that I am, because, I have this urgency, to be able to do more. And actually we have, we made a lot of progress the last 3 or 4 years. Just last year, I patented 11 different technologies in the field. And one of the major ones is what we call supercharged NK cells. Because, in the field, unfortunately, there's a lot of confusion as to, how we can strengthen the function of these cells. Some people have gone through the route of basically taking the cord blood and isolating what they call NK cells from the cord blood. And
the umbilical cord, right?
Umbilical cord.
Don't they usually do that for stem cells?
The stem cells too.
Got it.
But so that's basically, that's the hematopoietic stem cells that gives rise to NK cells. So that's the reason why they are taking it, because they think, okay, we're going to go to the progenitor cells. We're going to take the progenitor cells and we're going to develop that to become NK cells. And we've done that. We've studied this extensively, not only here, but also my collaborators in China. And we figured out that actually they behave very, very differently from mature NK cells. And they don't kill cancer stem cells the same way as, let's say, primary NK cells or supercharged NKs do. They don't actually, what we found that they don't kill at all, but they would produce some cytokines. Now, cytokines are very important for differentiation of the tumors. And so that aspect of it is very important too, except that when we compare, let's say, per NK cell function, what we are seeing is that supercharged NKs have about almost 50 to 100-fold more secretion of these cytokines than cord blood derived.
Meaning they're more powerful.
They're more powerful, exactly. Now, people make a lot of money off of these cells because, patients that get infused with cord blood derived NK cells, I'm sure on average they are paying anywhere from $50,000 to $100,000 because, the way they have to develop these cells takes a lot of different strategies and it's costly. Of course, they also make a great profit on it. But I believe that these cells are not going to behave the same way as NKs, and we have evidence to that, which we will be publishing pretty soon.
Wow.
So that's one area that the field is going. And unfortunately, again, we always go behind what is simple to do. we don't go behind what is effective.
Explain that.
So from the cord blood NK cells, for example, if you start from, let's say, 100,000 CD34 positive cells from the cord blood, you may end up getting upwards of 10 billion what we call so-called NK cells, which I don't, as I said, I don't believe that they are the primary NK cells. They don't function the same way. So the more cells you have, they think the better it is, because now you're going to be infusing the patients with these large numbers of cells.
Which is how they're selling you. We're going to infuse you with, 1 billion NK cells and you're going to be cured.
That's right.
Okay.
That's right. They don't realize that 10 billion NKs could equal to 100 million supercharged NKs. You know, so
and what's the killing ability of them? That's the thing that I've been learning with you. Yes. So I feel like our audience probably has never even heard much about the umbilical cord NK cells and all of that. I think newer moms might be exposed to this in some of the metropolitan areas. But if you haven't heard about this, it is a big thing that people are being told to keep the umbilical cord, take the stem cells from there, and then they save it. They store it and save it to re-inject you if someone gets sick.
Yes.
The child, the parent, anybody can use it.
Yes.
And you're saying that process is not effective because they're marketing you this idea and it's not 100%. It's not really effective.
Well, I was talking about the NK aspects of it.
So now, the stem cell aspect is a different thing.
Stem cell aspect is very different, depending on what they are isolating. If it's mesenchymal stem cells, it operates in a different way than what we call hematopoietic stem cells, which gives rise to the NK cells.
Got it. Good clarification.
So I mean, there are also problems with storing your stem cells, your mesenchymal stem cells, not necessarily, the cord blood. And I mean, during freeze and thaw, there's going to be a lot of problems with those cells. And so far, we don't have great evidence that those stem cells will actually heal something, There hasn't been studies where we can go behind and say, these are the cells that we gave and these are the cells that are there, because usually they disappear. And for good reason, because your NK cells, again, is not going to let too many cells to start populating your body, because the reason we are the way we are is because our NK cells look at each organ and say, well, this is how much it's going to be. It's not going to grow much bigger than that, Otherwise, we'll be, a spleen running around, So there's a lot of regulation that goes on, and we're discarding those things, and that's why we have to be very careful what we are doing. And I actually work with a stem cell biologist, a clinician who's freezing the stem cells, and he believes strongly that these are going to be very important in the future. And it may be, but I have not seen convincing evidence so far to tell me that, yes, this is going to be something very important. Or perhaps we haven't yet explored everything that we need to explore in the field.
On the natural killer cell side, though, you are seeing success.
With supercharged NKs.
So explain what supercharged NK cells are.
So this is different from, actually I was going to go to, but I can start with supercharged and then go back to induced pluripotent stem cell that also people are using in order to induce NK cells. And I know that there are some clinical trials that are happening in San Diego using those cells. In terms of supercharging NK cells, we don't go to the stem cells. We go to the peripheral blood. We take NK cells from the peripheral blood, and then we supercharge them by using strategies that we learn in the lab, and one of which is using a cell type called osteoclasts. Now, osteoclasts are in bone marrow and also in bone, basically, and they are very important for remodeling of the bone. But when we were doing studies on osteonecrosis of the jaw, and these are patients that are on bisphosphonates, bone-forming drugs, we found that actually these cells were, incredibly activating the function of NKs. And so even though we understood the biology of it in terms of osteonecrosis of the jaw, but that gave us also the idea that we can actually use these cells in order to expand NK cells. So the reason osteoclasts are working is because they are providing NK cells with all sorts of things that NKs need in order to expand. In addition to that, of course, we add our formulated, probiotic bacteria, because the way we formulated the probiotic is in such a way that it will actually keep your NK cells in a proliferative phase.
What does that mean?
It means that they will be dividing all the time. And it doesn't
making
kill.
more.
And making more.
Okay.
Right. And so the combination of those two strategies actually, gave us the ability to go from 1 NK cell to 5 million NK cells. And so we are very lucky right now. We can keep dividing these cells and keep injecting to the patient. Now, NKs are very different from T cells, because T cells, if you're going to be doing immunotherapy for cancer of T cells can cause graft versus host disease.
What's that mean?
Meaning that the cells that you're injecting could actually end up killing your own cells and cause disease, basically, and autoimmune disease, kind of. Whereas NK cells, no matter how many you inject, you do not see any evidence of graft versus host disease. And that's the reason why we said, okay, well, we're going to take allogeneic NKs, which means we're going to take NK cells that are healthy from healthy individuals. The reason being, when we went to the patients and we tried to supercharge their NK cells, unfortunately, we could not. We could not make them divide. We couldn't. They weren't strong enough, and they would die. So the
cells would die.
The cells, yes. No, not the patient, right? The cells would die, and so that defeats the purpose, basically. I know there are significant numbers of companies right now that they claim that they want to take patient NK cells and expand them and give it back to the patients. And I would say that I'm completely opposite, or I completely disagree with their view, because, again, we've shown that patient NK cells have several defects, one of which they cannot kill. The second thing, they cannot produce gamma interferon, which is basically the cytokine that actually drives differentiation of the tumors. More importantly, the gamma that they are producing is dysfunctional. So
which is why you have cancer.
Exactly.
But you're saying some of the patients' NK cells can't be supercharged, but some can, right?
If the person, yes, in a sense, you have to test them before you can even, decide whether that's the case or not. Most patients that we've seen, even stage 1 cancer, we do see some level of inhibition of their NK cells. So in a sense, if you want to have an effective therapy, I think the final way is going to go to someone's NK cell that it's completely healthy and can be supercharged and can be expanded.
And it doesn't affect the patient to have someone else's NK cells?
No.
Wow.
That was the whole reason. T cells, yes, but NK cells, we've been doing this with pediatric patients. We were injecting allogeneic, what we call allogeneic, taking it from, healthy patients and injecting in the pediatric patients. We have not seen any side effects of injection of the NK cells, So, I believe that the future, we're going to be actually looking into getting a bank of these cells from individuals that are very healthy, that their NK cells are functioning well, and that they are dividing well. So there has to be some screening that has to take place before we can actually expand these cells. In terms of the potential in the future, if you talk to T cell people, right now we're doing CAR Ts, in therapeutics. Now, CAR T is basically what we are doing. We're taking a patient T cell and we are inserting a different receptor. And that receptor interacts with, another ligand, which we call on the surface of tumor cells. And this interaction causes now T cells to become activated and kill the tumor. So that strategy actually started from UCLA. Several colleagues of mine were interested in doing those kinds of treatment. And now, this is a multi-billion-dollar, technology. The drawback to that is that patients end up going through what we call cytokine storm. And if they survive through cytokine storm, which sometimes they don't, because there has been situations where patients died from it. And the reason is just there's such a high level of activation of these cells that they are producing a lot of cytokines, a lot of factors that cause death of the cells rather than, and eventually death of the patient. And the efficacy of CAR T, not every patient benefits from it. The efficacy is anywhere from 10 to 20% at this point. And unfortunately, it can buy time, maybe a year or 2 years, but the cancer comes back. And it comes back with a revenge. Yeah. Because now they've down-modulated a lot of the receptors that we use in order to treat. And meaning that they decreased, and so we cannot do much of a treatment in these unless we now come up with another CAR T.
So it buys time, it just doesn't solve the problem. In fact, it hurts you in the future.
That's right.
Got it.
That's right. I mean, it buys you time, because otherwise those patients, they are terminal, basically. Yeah. They don't have any other choice other than going through the CAR T therapy. But again, it's not an effective treatment.
But the natural killer cell therapy with the probiotic is proving to be effective.
Yes.
And I want to focus on that because you also created the first humanized mouse model with an intact human immune system, which is why you're a gangster and a badass and why we want to celebrate you, because what that does is it almost guarantees replication in human clinical trials later. So it's like it advances the process. It speeds up the process. So when you have done that with the natural killer cell therapy and the probiotic, what have you seen?
Okay, so let me tell you a little bit about the humanized mice. So these are, we have a core facility at UCLA that makes these animals. And a few of my colleagues got together and started thinking about humanizing animals. And so it took a long time to get to a point, to where we are right now. And without giving you a long history of it, what we do with these animals, we actually take tissue samples, tiny tissue from thymus and liver, and we put under the kidney capsules of these animals. And then we also sort out those hematopoietic stem cells that we are talking about from the bone marrow. And then we are also injecting through IV. And we wait for 8 weeks to see whether they changed the immune system. And in majority of the cases, we do see that they changed. And then now those animals, what we call these are humanized, because they have human immune system.
So crazy.
Yeah.
Steven, are you flipping out in there?
I mean, I'm just processing.
Okay, continue. He's our science nerd in there. He lives for scientific stuff. So we can be very fruity and spiritual in here. And he's like, I need science. So I know this is very exciting to him. Okay, so great. Carry on.
Yeah, so then, we take these animals and then we implant them with human tumors. And what I've done is I've implanted with pancreatic tumors, as well as oral tumors. Oral because it's so simple to see it, whereas pancreatic, you have to open the animal to see. So we have now the two strategies, and we wanted to target cancers that are incredibly difficult to treat. And what we saw after injecting, so we implant the tumor and then we come back after 10 days. Once the tumor is established, then we inject them with supercharged NK cells. And what we saw that...
With the probiotic or without?
Both ways.
Okay.
And in both ways is very effective, but the probiotic helps actually the supercharged NK cells to remain very active and kill, So what we found is that the tumor shrunk almost to 10% of the original, tumor.
And how long after injection?
This is we've taken about 3 to 4 weeks.
Okay.
To sacrifice them. And the evidence, when we look at without supercharged NK cells, what we see that the tumors grow out of hand. I mean, there's a lot of stem-like tumors that are growing. But with the ones that we injected NK cells, all of the tumors are differentiated. And so we know differentiated tumors are very benign. They don't grow as fast. They are very easily killed. And so as a consequence, now the tumor is very small. We then resected the tumors from these animals, and we cultured them in the plate. Those that were taken from the animals that did not have supercharged NK, they grew very, very fast on the plate, almost like in 24 to 48 hours, completely, covered the plate.
Wow.
But the ones with the supercharged NK, it took almost 2 weeks to even get a tiny number of cells to start growing. Eventually, they will grow out because there are no NK cells remaining. Eventually, those cells, if we allow them for a longer time, when NK cells are gone, those tumors can revert and start growing. That's why we need to have NK cells all the time. We cannot just say, okay, well, we're going to inject one time and then we'll let it go. The tumors will shrink. But then eventually,
it's maintenance.
Yeah, exactly. So you need to kind of, have a schedule. And it will depend on the size of the tumor, of course, and the type of the tumor, So all of those decisions are going to be made based on studies that you would do with the patient. And we came up with 15 different tests that we apply to patients to understand the status of their NK cells and what we have to do. How do we have to treat the patients? Can we supercharge or not? All of these parameters will be tested, in the patient before we decide what we need to do. So in a sense, again, we're not shooting in the dark. We're not just putting something there and then praying God that something is going to happen.
I love that. Yeah. Stephen has a question. Go ahead, Stephen.
Sure.
I was curious, in your experimentation with the mice, is there several different groups where you have the control group where you don't do anything to it, but you have the tumor? And then is there a group where you kind of track the long-term, cure? Like, do you give it the supercharged cells and then give it chemo afterwards to completely kill off the cancer and then track long-term?
Yes. Yes, we have done those. And we've shown that actually chemo is a combination of the chemo and immunotherapy is what will eliminate the tumors. Because immunotherapy will differentiate the tumors and the chemo will in turn kill the differentiated tumors.
Interesting.
So that's one thing. And the second thing is that once NKs differentiate the tumors, now T cells can handle those tumors as well.
Is there a life cycle timeline that you have with mice that they live a certain amount of time and you can kind of scale that up to humans in terms of this treatment? Using these supercharged cells and using a plan that uses K cells mixed with chemo, can you create a different timeline for a human life based on how long the mice last afterwards?
Yes, yeah. Because again, in a mouse lifetime, we have animals that can survive from anywhere a year to 2 years. And so we can scale it on that level. But remember, we're dealing with humanized mice, and these animals are so precious and so incredibly difficult and expensive to make that we don't have the luxury of, doing this in many different directions. So we have to prioritize in terms of what is more significant for us to look at. And,
so that's why you use the pancreatic and the more aggressive cancers to test it.
That's right. Yeah. I wish, we had an army of researchers, which, I'm trying to train them, and so they can get out and do the same things. But at this point, because of the difficulty of the model system and also the amount of money that it requires to do these kinds of work, we are limited as to what we can look at.
How much does one of those humanized mice cost?
Well, Jackson Laboratories were selling it for $2,500 each.
Wow.
And this is not even considering doing the experiments, because with the experiments, let's say, if we're going to be doing 1 liter of experiments, and it may cost anywhere from $70,000 to $100,000.
Wow.
Yeah.
And how many mice do you need to have control group and to have enough data to actually give you an answer?
Usually we take anywhere from 10 to 20 animals per experiment.
And do mice have a similar blood-brain barrier that humans have? Is that why they work? Okay.
Yes. Mm-hmm.
Great question, Stephen. I wonder, have you guys tested glioblastoma on these humanized mice yet?
That's coming.
Okay.
So I actually brought 2 experts from Europe to stay in my lab for a year and a half to establish the model system. And we're working actually with Dr. Nathanson at UCLA, who's a GBM specialist. We're working with him in order to be able to get these tumors into humanized mice, and then try to study the same way as we did with the pancreatic and oral. Although GBMs are different, they have completely different processes. For example, one of the things that we published on glioblastoma is when supercharged NK cells interact with pancreatic tumors, it basically would eliminate, and there is no excess pro-inflammatory cytokines that gets made. With GBM, we found very different, because supercharged NKs can eliminate them, but at the process of differentiation of those cells, now differentiated tumors actually trigger pro-inflammatory cytokines. So that's why these patients have even further difficulty, because they have to go on dexamethasone, or they have to go on a lot of steroids to keep that inflammatory process down.
And that inhibits the immune system.
That's right.
Which means it's inhibiting the NK cells.
Yes.
That's so interesting. Yeah, that's where we've been at with my mom. We had to get her on the dexamethasone, unfortunately.
Yeah. It's unfortunate, because we're trying to understand how we could target that without targeting the immune cells, the NK cells. And it's, one of the problems with also GBM is that once they become differentiated, it's not the end of it. They can actually go back to stem-like. The differentiated tumors then go back to stem-like. So it's almost like they are ping-ponging between the 2 states. And you try to catch them to push them to differentiation, but then they can come back again and see the tumor,
Little assholes. They won't let us catch them.
I know.
I wonder, so you're working on the GBM, but in terms of the other terrible cancers that you've tested, pancreatic being one, what have the results been like?
In mouse models?
In the humanized mouse models?
Yeah, quite successful, because these animals are doing very well. The tumor shrinks in a great level.
Has it been eradicated at all in any of these mice?
They don't grow. I mean,
they stabilize.
There are tumors there. Well, because we could see it, but we cannot grow them as well. We could see the tumors that are there, but they are just frozen.
And is pancreatic cancer the deadliest?
Yes.
Yeah.
Right.
And so the fact that you have in humanized mice with humanized immune systems figured out the combination with natural killer cell therapy with your probiotic therapy, figured out how to shrink and stabilize pancreatic cancer, what would that do, in your opinion,
in
humans?
Well, we believe that we're going to see the same thing, that we're going to shrink the tumor and also we're going to prevent the tumor from metastasizing.
And extending life.
Yeah, exactly. And again, remember, the tumors that are metastasizing are the stem-like tumors. Those are the aggressive tumors. So if NK cells are capable of eliminating those, then of course there's not going to be any metastasis. So basically, even if there are some differentiated tumors there, they're not going to grow. They're just going to stay there, and they're going to be frozen, basically. Or alternatively, your T cells will target them, or chemo will target them. So,
but would you have to use chemo as well, or would natural killer cell therapy and probiotic be the future?
I think, we hope that we are going to find, at this point, in terms of chemotherapy, we believe that because, immunotherapy can be backed up by chemo to kill the differentiated cells, I think that's a strategy that one can have right now. Certain chemos have also been shown to actually strengthen the immune system. So,
which ones?
So, for example,
Temodar?
CDDP, which is cisplatin. We've seen that cisplatin can actually activate the function of NKs. So it's a double-edged sword, basically.
Yeah. What about Temodar? My mom's been doing Temodar.
Yeah, we haven't yet studied Temodar, so we are going to be starting with it too, because we're going to be studying GBM. So that definitely is going to be one of the factors that we're going to be testing. But because of this double-edged sword process, we're thinking, what if we come up with immunotherapeutics that doesn't kill the immune cells, and in
the
turn,
good cells,
exactly, and in turn can strengthen those cells,
which is what chemo does. It kills the good and the bad.
That's right. At certain stage, though, you have to look and see what stage of disease it is, but in general, the fact that there's a lot of toxicity with chemo is at some point we have to change it, because we don't want patients to go through,
of course,
the toxic treatments.
But why would someone with pancreatic cancer, hypothetically, need chemo or any other treatment if the NK cells and the probiotic is working?
Well, as I mentioned, NK cells target cancer stem cells. Once the tumors are differentiated, NKs no longer deal with it. They don't touch them anymore. So now the T cells have to come in, and we have to have other strategies to bring in. Now, that doesn't mean that we cannot manipulate the system in order to make NK cells to kill differentiated cells. But that's going to be another strategy, which will come up with like biological treatments that we do, like, for example, anti-PD-1, anti-HER2/neu, because these antibodies, when they bind to differentiated tumors, NKs now kill them in that fashion, but they cannot kill them directly.
Got it.
So things are a bit complicated, but I think we have very specific pathways that we are taking, and we believe that at some point we're going to find every single strategy that we can use in order to eliminate tumors without the aid of chemotherapy.
I love it. Steven's got another question.
So I'm curious, do the K cells require a strong immune system to actually work? Because I'm curious about how this treatment could be applied to like leukemia when they already are like immunodeficient. And that's not really a tumor-based cancer. It's more like everywhere. So could that be a viable treatment for things like leukemia as well, or no?
Yes, every single cancer. The reason actually we have cancer is because our NK cells become disabled in the first place. And to give you an example, one of the projects that I was very much interested to understand was what happens to the NK cells pre-tumor, induction. And so in order to address that question, I worked with a couple of colleagues at UCLA they had a mouse model where there was KRAS mutation in the pancreas. So this is one of the major mutations that induces pancreatic cancer. So we took those animals and we divided them and gave them different kinds of diets, like high fat versus low fat diet. And then we came back and asked, how about the NK cells? What happens to the NK cells in these animals? All the animals that had KRAS mutation and that were fed with high fat calorie diet, their NK cells was completely wiped out. That basically their NKs were dysfunctional. And those were the animals that first started showing the signs of cancer. Then, of course, the animals that had the KRAS mutation and had also a healthy diet, they came up with the cancer next. And of course, the healthy animals with no mutations, they never had any issues with pancreatic cancer.
Ooh, Stephen's raising his hand. Go ahead,
Sure.
Stephen.
You know what
I feel like you should be up here next to me.
Yeah, sorry.
This is your day.
You know what I'm really curious about, actually, based on what you just said? Has there been, and I don't know if we have data going back 30, 40, 50 years on NK cells, has there been a trend towards processed foods and different things like that leading towards human beings not producing as much? Like, has it been an evolutionary thing that we don't produce as much now as we did 100, 200 years ago? Have we kind of lost our natural defense versus cancer?
Yes, I think that's an excellent question. And I believe that's one of the reasons why more and more we're becoming immunosuppressed. We're basically immunosuppressing ourselves with the environment that we are in and with the diets that we have. And that's partly because we're consuming a lot of sugar, we're consuming a lot of fats, different kinds of fats, a lot of chemicals that are being added to our foods in storing them, increasing their shelf life, all the additives and fillers and everything else that is being added. So I firmly believe that those are contributing to. Now, the way they are contributing is that because they are targeting our gut microbiome. So they're basically destroying gut microbiome, and as a consequence, now our immune cells cannot be regulated in the proper way.
See, remember when we read Jane McClellan's book, How to Starve Cancer, and she talked about how she used to think that bloating and diarrhea was a normal thing. And that's when she started to realize that the microbiome being compromised was the first step in cancer. Because the body is so out of whack. And that's such a great point.
No, that's a fact. It's not a fiction anymore. I mean, we know that gut microbiome, it gets screwed, we have cancer as a consequence.
Wow. So I have a couple other questions as we're getting close to the end. What are your thoughts, because I know that you've been working with Tracy.
Yes.
What are your thoughts on the effects of marijuana on cancer?
Right.
And on natural killer cells?
Right. I never thought I would become interested in the subject because there's a lot of important, subjects to study. That doesn't mean that, this subject is not important. It's just, I wanted to pick and choose what I wanted to study until, Tracy came to me and asked me whether I could grow Sophie's tumors. And
Sophie,
then
incidentally, is Tracy's daughter with brain cancer.
Yeah. And so we started looking into it. We got the tumor. We started, growing. And I could see that these tumors were growing incredibly fast. And so I started thinking, okay, what is going on? Why is it that I'm seeing such an incredible growth in the plate, but yet we are not seeing the same rate of growth in Sophie?
Is it because it's exposed to air?
I think it's the regulation that is taking place. I think also the cells, different types of cells that interact with the tumor. We're still trying to understand the interactions, but so one of the things that was different in Sophie was because she was on cannabinoids. She was on marijuana since 8.5 months old. So that put me to think, or made me to think, okay, what is going on? Is this something that is contributing to slowing down of the tumors? And so I asked Tracy, I said, I'd like actually to look at the immune system of Sophie and also patients similar to Sophie that are on cannabinoids. And interestingly, what we found was that patients that are on cannabinoids, their NK cells are not dysfunctional, which is basically, I think that's one of the reasons why these patients are benefiting from it. We're still trying to understand the pathway, mechanisms, because if you read in the field, everyone suggests that there is immunosuppression with cannabinoids.
Suppression meaning bad.
Exactly. We are challenging that notion right now because our studies are not supporting a suppressive effect of cannabinoids on immune system. So we're the jury is still out. We haven't exhausted everything, but in our preliminary experiments, we don't see immunosuppression. We see actually stabilization of the NK cells.
Which is interesting. So, and this leads me to my mom, because I teased earlier that we were going to talk a little bit about my mom. You studied her NK cells back in March of this year.
Yeah.
And you said this is why your mom is doing well. She has an abundance of natural killer cells. The killing cell ability was high, which was very shocking because when you normally look at cancer patients and she has stage 4 brain cancer, glioblastoma, her NK cells should be compromised.
Yes, right. Well, that's something that, I'd like to discuss because if you remember, I mentioned also that even though she has good killing activity, the gamma interferon is a bit, less than I would expect.
In March or the new one?
In March.
Okay.
Oh
yeah, I do remember that.
Yes, and that was the reason I suggested. Okay, let's see, whether if she goes on probiotic bacteria, maybe it can boost up, the gamma induction. And so that was concerning to me because that's the profile that I also see in recurrences. And now I believe that could have been the reason why your mom recurred with glioblastoma.
And in fact, that was March. If you look at her MRI in July, they missed it. You could see it starting to form, and they missed it.
Right.
So we didn't see it until September.
Yes.
So it was very close. So your theory makes a lot of sense. And before March, she had been on marijuana consistently up until that October. In October, we stopped the marijuana because she was going overseas to my wedding. And after that, we wanted to have a little bit of normalcy because she was literally just out cold all the time. She's very sensitive. So the marijuana was really just making her sleepy.
Yes.
So she might have had some of that beautiful effect of the marijuana on her killer cells.
Yes.
And then so you retested her, obviously, a couple weeks ago when we knew she had the recurrence.
Yeah. So there are this is something that I may need to talk to you, and I don't know whether you want me to discuss.
Go for it. Yeah. It's okay. Okay. We're very open here.
Okay. So what we are seeing is that again, incredible activity, but the numbers are low now.
Is it below 0.7 in the killing ability?
No, the killing is okay.
Okay.
Even the cytokine secretion is okay. It's the numbers of the cells per se is low. Because from 30 cc's of blood that we got, we recovered basically 2 million cells, which is very, very low.
Where was she at in March in terms of cells?
We were recovering almost 100 million.
Wow.
Yeah.
Wow.
Yeah.
So she's depleted.
She is depleted. And I'm trying to understand why. I know she went for NK therapy, and she got NK therapy.
She did.
And
we added the probiotic every day.
Right. And also she, did she have any chemo?
She just started it this last 5 days. I mean, like she's just finished a couple days ago.
Okay, but she wasn't on chemo when I took the blood.
No, she did do the Keytruda at that point, the immunotherapy.
That could have been the reason too, because there's a possibility that there's something called activation-induced cell death. That if we super activate the cells, then they will commit suicide. So, I mean, this is just a hypothesis.
Yeah.
It doesn't mean that, that's the case or not. I have to study her. Hopefully, I'll get a little more blood to understand whether she has stabilized now or, but that was very concerning to me because we tried every which way, to recover more cells from her, but unfortunately we couldn't. And I think, it's possible that, even the NK therapy that they, gave had something to do, with the process. We just need to, because the reason I'm saying is because there was another patient who went to China and received, approximately 40 different cell types, not supercharged NKs. He did not receive supercharged NKs. And when he came back, when I tested him, he had exactly the same process.
Wow.
He had improved, the function had gone up, but the numbers came down significantly.
Wow.
So it could be that this is something to do with the treatment.
So maybe when they're injecting you with the NK cells, they're just committing suicide. It's actually doing the reverse.
Yeah.
It's hurting.
Right.
Well,
at this point, we don't know whether it's, helping
or it could be
hurting.
It could be the immunotherapy too.
Right.
So is there a way to take healthy NK cells and inject them into her?
You have to manipulate those NKs. If you take, let's say, this is where the problem is because FDA doesn't let investigators to modify cells. And if you take NK cells the way they are without modifications and inject them to the patient, what's going to happen is they're going to become inactivated because, the microenvironment in patient is suppressive. So it's going to suppress it because they don't have any defense. So I think eventually, FDA has to change their minds and let people start actually modifying these cells, like, for example, supercharging these cells and injecting based on the evidence, I think just injecting NK cells directly, it's not going to do anything.
Yeah. So what would a potential solution be, in your opinion?
Maybe transfusion even.
Blood transfusion?
Yeah, bring more cells in, Try to boost, the bone marrow, get some more cells, Lunesta, for example. Patients that are on chemo, they usually receive, when, their cells deplete, they usually get a booster shot with Lunesta because that also gives, increases it stimulates the bone marrow to actually bring in more cells. So I mean, that could be a strategy. I'm not sure whether her doctors had tested her and seen whether she had lower levels of lymphocytes. I'm interested to look at those results. And determine basically whether she has any kind of immunosuppression.
Got it. Yeah, well, we will test that for sure.
Yeah.
I wonder, this may be a reason to put her back on the marijuana because I had stopped it for a little while.
Yeah.
Might be a reason to bring it in and see if it can boost it.
Yes. Right.
Right?
Yeah. Yeah. Yeah, because we are seeing depletion. Why it's depleted, we just have to now find out why.
Yeah. Interesting. This is so fascinating. I mean, I know there's so much still to be done, but you've made so much incredible progress as well. And so are you in the clinical trials now for the pancreatic cancer natural killer cell probiotic therapy?
So what I mentioned, FDA is one of the biggest hurdles right now for supercharged NKs. That's was part of the reason why I actually took the technology and went to China, because I thought it was going to be much simpler to do it there, and it was, and we did inject to a patient, and the patient survived for the tumor went away, and for a whole year the patient is doing very well without any problems. But because of the relationship now with China, it's almost everything has halted. The communication is not there anymore, and and there are difficulties.
So where else can we go? Germany?
It's difficult. My hope is
the Bahamas. I feel like they would allow it.
There's lots of people who want it, but I kept thinking, I need to have control over how things are being done. And if something is being done in China and I'm not there, I don't have a control over it. So my job now, with the two companies that I'm actually working to establish, Encore and NK Immunity, is the possibility of not only having a GMP to produce these cells, but also putting it in the patient. And my hope is that I'm going to accomplish this within maximum 9 months. So that's the direction I'm taking right now.
Yeah. Well, we're all going to help you in that, because we need you to build this lab so that we can get these things going, because we all need it.
Thank you. Yes, I think it's extremely important, and that's why I established Cure Cancer Now Foundation, because we have this urgency. That's why Cure Cancer Now.
Yeah.
We don't want to cure this in 20 years or 50 years. We want it now.
How can people support Cure Cancer Now?
They can go on the site and hopefully, support our effort. They can also find me at UCLA site, because we have a giving site at UCLA, and they can support in that way as well. And I always welcome people to come to the lab and see our operation and talk to us and talk to my people in the lab and find out what exactly we are doing, and hopefully they will join our team, and so we can defeat this damn disease.
The lab is so cool. Steven, you should go check it out. You would live for it.
Can I?
Yes.
Of course.
It's so fun, and it's just great to see people working diligently and constantly on finding a cure for everything that we're suffering from. I want to ask you our final question, Dr. Jewett, we always ask is, because the show is better together, how are you getting better in your daily life? Are you meditating? Are you eating cleaner? What are you doing specifically?
My diet is very bland, meaning that I don't intake any sugar. I don't intake any gluten. I don't intake any saturated fat. I stay away from, the only meats that I will eat would be fish and chicken. A lot of vegetables, a lot of greens. I stay away from sugary fruits as well. I try to optimize my diet based on greens and vegetables, and less of everything else. But unfortunately, my lifestyle is very stressful, because I cannot change that. The stress is there, and I try to, by taking walks, by hiking, by trying to maybe shut down my brain at times.
You actually do that?
I try, but
I've been on the phone with Dr. Jewett at like 10 p.m. at night, and if I let her, she would go till 1 a.m. explaining everything. Like, you are so fierce and so dedicated. So I'm glad to hear that you do try to shut your brain off at times.
And I mean, the last thing I would say, unfortunately, I drive my people incredibly hard in the lab, because the minute they come and tell me, oh, can I have, let's say, this weekend off? And I say, well, the patients don't have weekends off, and we need to come up with something, So I know I put a lot of pressure on my people, and the same way I put pressure on myself. But I think we need to have dedicated people that will do this, because otherwise this disease is incredibly difficult. And we need to learn more, we need to understand more, and we need to take action. Inaction is not acceptable with cancer.
No. And we're better together, so you need your team.
Yes.
Dr. Jewett, thank you so much. All right, Steph, what have we learned since Steven's not here? He had the runs, guys. So Steven's unable to join us for...
Oh my God.
I love spreading rumors. No, Steven is not able to be with us for this moment, but it's not because he has the runs or anything else that could be horrible.
He had to run.
He had to run.
But not the runs.
Exactly. So, Steph, what have we learned?
First and foremost, I don't know how you get connected with the most badass people ever. But Dr. Jewett is so amazing. I think the biggest thing that I learned was that it's confirmed that when you have stomach issues with your biome, it is
your microbiome.
It is always like the first indicator of a person who is developing cancer, who has all across the way, patients who have cancer have a microbiome issue. That's crazy.
And we talked about it, maybe a month ago or something. And, it isn't a coincidence when things fall into your lap. At least for me, it isn't, because I'm really watching all the connections. So I thought that was really fascinating as well. I mean, there's so much to learn from Dr. Jewett, and hopefully her research gets funded quickly so that we can get more of this out there.
And on our after show, I'm so excited to ask you about everything you did with her after, because I know you were able to bring your mom to see her, which is amazing.
Yeah. So tune into the after show, guys, which is part of our Patreon community. So if you haven't joined our Patreon community, please do. We really want everyone who listens to this show to join it so that we can make the show completely ad-free, and also to build a larger community. We have a lot of big plans and a lot of exciting things we want to do with you guys, because you're our tribe, you're our peeps. And so the more we get connected, the better. So if you'd like to join us, you can go to bettertogetherwithmaria@gmail.com, and we can send you the link. But also, you can just go to patreon.com/join/Maria Menounos.
And in all of the summaries of everything we do, you will see that link.
There you go. That's even easier. In the meantime, thanks for joining us as always. Thank you for supporting the show and for rating and commenting and subscribing. The biggest gift you can give, I think, at this point is to share it with people, share it with your friends who need to know this information. Everybody needs to know that their gut microbiome is important. Everybody needs to know about a lot of the stuff that we're doing on here. That's why we're doing it. So share it with a friend, social it out so that more people can become educated on all of this. If you want to visit and support Dr. Jewett's Cure Cancer Now Foundation, you can go to curecancernowfoundation.org. And in the meantime, you can follow us at Maria Menounos, at Steph Sabra, at Stephen Lemieux Photo. And remember, be nice people, make good choices, and be present.